Hello everybody, and welcome back to my basement. Before we get going today, a couple quick notes. First thing is we are in my basement. It is the middle of the day. My neighbors, I can see them through a window right here, are building a shed next to their house. There is a very small child kind of up that way. And there's also a dog that gets jumpy when the mail gets delivered. So there may be some noise in the background for this one. Sorry, that's just kind of occupational hazards at this point. Second thing is that starting the day this becomes available to you, which is Thursday of this week. That's Thursday the 12th or 15th, Thursday, October 15th. I will be taking over the review sessions from Mr. Merchant. So just be aware of that. My I'm disappointed that I had to miss the beginning of the semester because now I'm jumping in halfway and I feel like I don't know anybody because I don't know any of you. So starting again, it's Thursday, October 15th, which is when this becomes available. Come to the reviews, come say hi, introduce yourselves because I want to meet all of you because I'm going to be with you not just for the rest of this semester, but also all of next semester. So come say hi so we can get to know each other a little bit. So that stuff out of the way, let's get started. Our topic for today is a really fun one. We're going to talk about chromosomes today. And just like last time, we have three parts here. So first, we're going to talk about the chromosomal theory of inheritance. And we're going to look at another biologist who kind of more or less accidentally got involved with genetics named Thomas Hunt Morgan. So I want you to be able to sequence his experiments and tell me what they showed, be able to explain what he found and what it means. Then we'll look at the inheritance patterns for sex chromosomes specifically. So compare and contrast, and HD, that's hypothesize and diagnose, sex linkage. Basically, if a gene is on a sex chromosome, how does that affect its inheritance pattern? And then finally, we will look at violations of independent assortment. Now, last time I mentioned that Mendel didn't quite have this perfectly right because he didn't know anything about chromosomes or linkage or anything, but we know about that now. And when things are linked, that messes with Mendel's idea of independent assortment. So we're going to talk about that in some detail today. So sequence and hypothesize and diagnose the patterns of inheritance, basically the linked versus unlinked and that kind of stuff. Now, before we start, I just want to kind of orient us in this broad set of material. I want you to consider this set of lectures as a unit. You started here with mitosis and meiosis, basically looking at chromosomes and how they separate, ultimately making gametes and then recombining into the offspring. Without really getting into what's going on there, you just kind of did the nuts and bolts of the processes. Then last time we talked about Mendel and genetics, so that sets the basic rules of the system. Today we're going to apply those rules specifically to chromosomes. So we're going to take kind of the two threads that we've done already and put them together. Next time we're going to talk about how it's the DNA in the chromosomes that matters when it comes to inheritance. So we'll look into things like the DNA structure and how it actually works to make proteins. So to review a little bit, we've previously talked about meiosis and we've also talked about genetics. So we have this process, right, where you have meiosis, the allocation of chromosomes from a diploid parent cell into the haploid gametes. And we talked about Mendel and how he talked about how heritable factors were passed on from parents to offspring. Now you had these kind of two things happening in parallel where Mendel figured out the rules but not the mechanisms. Cell biologists were looking at the mechanisms but didn't realize that chromosomes were related to the rules that Mendel had figured out. That all comes together in the early 1900s with the chromosomal theory of inheritance. So let's start with that theory. There are two main components here. The first is that genes have specific loci on chromosomes. Loci is the plural of locus. It just means location on a chromosome. And chromosomes are the things that undergo segregation and independent assortment. And this is where we differ from Mendel a little bit. So Mendel said that genes, heritable factors, are the things that undergo segregation and independent assortment. What we now know is those rules are correct, but they apply to chromosomes rather than individual genes. So how did we figure this out? This is what we want to address today. Why isn't it the case that each individual gene is behaving independently during meiosis? This is what we're going to figure out. So we'll start with Thomas Morgan and Drosophila melanogaster, which is the fruit fly. Now Morgan, as you can see right here, this is Morgan, played by Peter Dinklage, and he was a biologist in the early 20th century, and he was actually an embryologist. He wasn't a geneticist, at least not at first. Basically what was going on is that from the 1860s into the 1900s, cytologists, cell biologists, so cyto, you should always think cell when you see cyto, that just means cell. So cell biologists were looking at chromosomes and seeing that they behaved the way that Mendel's heritable factors behaved. Now, Morgan was originally a skeptic of Mendel's work and of chromosome theory, the idea that chromosomes would be involved in inheritance. But Morgan ended up coming around to the view that chromosomes were the thing for inheritance, and he actually provided the first experimental support that chromosomes are the things that carry genes. And basically he showed that chromosomal theory fits what was known about Mendel's heritable factors. And here, this picture, this is not related to anything. This is just actually pictures from cell biologists, hand-drawn from the 1800s, looking at like chromosomes and different types of cells during mitosis. So you can see right here, like that's a cell in what phase is that? It's anaphase right there, right? Here you got metaphase, and it's just these really nice hand-drawn kind of cells, and if you search Walter Fleming, you can find more like that. So the model organism we're dealing with today is Drosophila melanogaster, which you all know as the fruit fly. If you've ever left bananas out in your dorm or in your kitchen for a little bit too long, you have seen these little brown flies with bright red eyes buzzing around your room. Now, as a model organism, fruit flies are great. They have lots of offspring. They have a very short generation time, about 2 weeks, and you can actually modulate with that with temperature to make that even faster. The other thing about Drosophila is that even back then we knew what their chromosome situation was. So we know that Drosophila has 3 pairs of autosomes and 1 pair of sex chromosomes, which in their case is either an X or a Y, similar to us, even though the system works differently. Now, when we describe traits in Drosophila, we're going to use slightly different notation than you've been familiar with so far. So we need to introduce the term wild type. Wild type just refers to the most common phenotype for a character in natural populations. For example, in fruit flies, red eyes are the most common eye color. So we call red eyes the wild type phenotype. Now, it's very important to note that wild type does not mean dominant. Some wild type traits are dominant, some wild type traits are recessive. It depends on the specific trait. And I will give you an example. I want everyone to look at your hands real fast. Everyone look at your hands. I'm imagining most of you are looking at hands that have 5 fingers on each hand. Well, that is the wild type trait for humans, having 5 digits on our hands. But the dominant trait is to have 6. It's called polydactyly, and there's lots of forms of polydactyly, but one particular form of polydactyly involves having an extra little finger right here next to the pinky. It doesn't have any bones, and you can't do anything with it. But that trait, that form of polydactyly, is dominant over having 5 fingers. So wild type, 5 fingers, dominant, polydactyly. Now, by the way, some of you might look at your hands, and you might see a little scar right there. If you have a little scar right there, that's what happens because they cut it off basically at birth because there's no point to having it and it causes no harm to get rid of it. So probably some of you watching this will be like, oh, I've got that little scar right there. That means you carry the trait for polydactyly, or you don't carry it, you have the trait polydactyly, and that finger was removed. Fun fact. And now everyone, if we were in class, everyone right now would be going like this, but we're not, so can't see that. Now, the alternative to wild type traits are called mutant phenotypes. These are the alternatives to the wild type, and for our purposes today, what we care about are white eyes in fruit flies. Now, the notation is also going to be a little bit different from what we did previously. Now, last time you may remember we just used capital and lowercase letters. where the capital letter matches to the dominant trait. So for flower color, we use purple and white, capital P for purple, lowercase p for white, because purple was dominant, white was recessive. For Drosophila, it's a little bit different, and this is the case. Every kind of model organism has their own system for notation for the different traits. So for Drosophila, the symbol for the gene is based on the first observed mutant, not the dominant trait, which is confusing. So for eye color, for Drosophila, the allele for white eyes is W, and the allele for red eyes is W+, where the plus indicates wild type. I know that's confusing because it's different from what we just did. Sorry, that's just how things are. Biologists are really bad at being consistent when it comes to stuff like this. Every different group has their own system, and that's just the way it works. Sorry. So again, the symbol is based on the first observed mutant, and then if you have a plus, that indicates the wild type. So now what we want to do is correlate the behaviors of the alleles through genetic crosses and the chromosomes through meiosis. And in order to do this, we're going to do a cross, just like Mendel did, but we're going to do it with fruit flies. So our P generation here is going to be red-eyed females crossed with white-eyed males. That's our kind of true breeding P generation individuals. Now, when we do this, the F1 all have red eyes, similar to how our F1 flowers were all purple when we talked last time. So this first tells us that red eyes are dominant over white eyes. Great, that's useful information. We know that red is dominant over white. When we do our F2, we do it the same way we did before, where you take your F1 generation, a red-eyed female, cross her with a red-eyed male. And in the F2 generation, just like before, we get a 3-to-1 ratio of red eyes to white eyes. Perfect. Everything is working exactly like before, except there's a wrinkle. There's always going to be something more fun here, and it's this. All those white-eyed flies were male. There were no white-eyed females in the F2 generation, and that's weird. So now we have to explain that. The explanation for this is how sex determination works in fruit flies. Now, fruit flies are different from humans. In fruit flies, the Y chromosome does not determine the sex. The Y chromosome contains the genes to make sperm. I should say contains the genes to make sperm, but it doesn't determine sex during development. What determines sex is the ratio of X chromosomes to the sets of autosomes. And in a second here, we're going to abbreviate sets of autosomes to the letter A, right? That just means sets of autosomes. So that ratio is what determines the sex. So if you have XX for the sex chromosomes and then 2 sets of autosomes, that's a 1-to-1 ratio, which makes that individual female. If you have XY and 2 sets of autosomes, that's a 1-to-2 ratio. That gets you a male fruit fly, and these are the males right here, the smaller with the darker abdomens. If you have XO, so you have an X chromosome but no Y chromosome, that ratio is 1-to-2. That's going to be a male, but no Y chromosome means they're sterile. They can't make sperm. And we can keep going. You could have XXX to 3 sets of autosomes. That's a 1-to-1 ratio. That's a female. And you have XXY to 2 sets of autosomes. Well, that's a 1-to-1 ratio of X chromosomes to autosomes. That's a female who I think in some cases are capable of making sperm. It's a little bit confusing, but you end up having the female phenotype in that situation because, again, the Y chromosome is not determining sex. So that's how sex determination works in fruit flies, and this is going to be important for explaining why all those white-eyed flies in the F2 were male. So why were there no white-eyed F2 females? Well, the eye color gene is located on the X chromosome, and there's no corresponding locus on the Y chromosome. So let's work out this cross, and we're going to see exactly how chromosome theory, which is to say the gene being located on the chromosomes, specifically the sex chromosomes, explains our observations. So here is our cross. So this is our P generation, we're going to go through a monohybrid cross, starting with the P and working it out from there. So we have our red-eyed female, right? XX, W plus, so that's red-eyed female, true breeding, and white-eyed male. Okay, X, W, Y. There's no corresponding eye color gene on the Y chromosome. So now in F1, we cross these, you're going to get females that are heterozygous, right? XW plus with XW. And over here, you get red-eyed males that are XW plus with the Y, right? Those are your only options there. Now in our F1 cross, here are the individuals, our heterozygous females with our red-eyed males. This is what's going to get us our interesting results. So let's work this out. The first thing is we could have an XW plus donated from the male, and that can be fused with an egg that has XW plus from the female. So that gets us homozygous dominant, or red eyes, and that's going to be a female with red eyes. Down here, you're going to get another female, heterozygous, which means also red eyes. Now over here, you're going to get a male, right, Y chromosome, with a dominant eye color allele. So that's going to be a male with red eyes. And then down here, this is the interesting one, you get your Y chromosome and your recessive eye color allele. So that's going to be white eyes, male. In other words, the only place where you find white eyes are in males. So it preserves the 3-to-1 ratio of red eyes to white eyes that you'd expect, but all the white eye individuals happen to be males because of the way these alleles are located on the sex chromosomes. So Morgan's findings were that specific genes were carried on specific chromosomes. He was able to document this by showing the unique inheritance pattern for genes specifically on the sex chromosomes, the X chromosome in this case. The fact that the behavior of the alleles corresponded so closely to the behavior of the chromosomes was extremely strong support for the chromosome theory of inheritance.